Hello and welcome to Teach Me in 10. In this episode, I speak to Dr. Tara Crawford-Parks, who is the Director of R&D at DNA Genotek. Tara introduces the HEMAcollect protein blood collection tube, developed by DNA Genotek and designed to enhance sample integrity for proteomic research through its proprietary ProteoPrecision technology. She'll also discuss the common challenges of pre-analytical variability in proteomics and how the HEMAcollect protein blood collection tube addresses these issues. Let's get into the video. Hi Tara, thanks so much for joining us today. I think let's start with the basics. So what is proteomics and what are some of its applications?
Hi, thanks for having me today. I'm excited to be here and talk about this topic with you. So proteomics is the study of proteins and their functions, and it's a rapidly advancing field offering insights into biomarker discovery, clinical diagnostics, drug development, and disease research. Blood plasma is a common sample type used to study proteomic profiles in individuals among various biological samples. And the plasma fraction is typically isolated from the whole blood and widely used in proteomic studies due to its accessibility and the ability to reflect systemic states.
Right, and so what makes blood collection and handling such a challenge for proteomic research?
Traditional blood collection methods for proteomic research face considerable challenges in the pre-analytical stages of sample collection and handling. So these include protein degradation, hemolysis, platelet activation, and the release of intracellular and platelet-derived proteins. These concerns do necessitate immediate plasma isolation and freezing, and it does have the potential to compromise that downstream sample quality. Delays in the sample processing steps or non-standardized workflows across multiple sites, as well as the introduction of temperature fluctuations during storage and shipping, can introduce significant variability, which will have a compromising impact on downstream data quality and interpretability. Researchers predominantly do use general blood collection tubes like EDTA tubes for plasma-based proteomics. While these are established and widely available, these tubes really weren't designed to stabilize proteins, and they lack extended room temperature stability for proteins and require that upfront immediate processing. So the scientific community broadly recognizes that there is a need for solutions that maintain protein stability at draw time concentrations, which really are enabling minimal pre-analytical variability and streamlining the sample management for proteomic studies.
I see. So what inspired the development of a new blood collection tube for plasma proteomics, and how does it address the challenges you mentioned?
To overcome the critical pre-analytical issues that I outlined, DNA Genotek developed the HEMAcollect protein blood collection tube, which is powered by the novel ProteoPrecision technology. And this proprietary liquid stabilization solution was designed to stabilize plasma proteins at the point of collection, minimizing proteomic profile changes, proteolytic degradation, hemolysis, and the release of the intracellular and platelet-derived proteins. The HEMAcollect protein BCT was also designed to stabilize proteins, ensuring a high-quality sample while providing that extended preservation of the plasma proteins for up to 7 days in the whole blood at ambient temperatures to support flexibility in plasma isolation workflows.
So how was this new blood collection tube tested, and what are the key performance indicators you analysed?
To evaluate the performance of this blood collection tube, studies were conducted where paired venous blood samples were collected by study participants into the HEMAcollect protein BCT and compared against general EDTA BCTs. In these studies, we broadly would isolate plasma at baseline or T0 within 2 hours of collection, and then subsequent time points were taken involving plasma isolation from the EDTA tube or the HEMAcollect protein BCT, and then the plasma was isolated at those individual time points, including up to 7 days at room temperature, and through simulated transport conditions, which ranged broadly from 4 degrees Celsius to 30 degrees Celsius for a whole blood sample, and minus 10 degrees Celsius to 30 degrees Celsius for an already isolated plasma sample from the whole blood. So some of the key things that we looked at in these studies, we evaluated hemolysis. So one of those key things that we mentioned were could have an impact on that pre-analytical workflow. So we did both a visual assessment of HEMAcollect protein BCTs to look for a change in or red color of the plasma, which is indicative of hemolysis, as well as doing a more quantitative measurement of hemoglobin, where we demonstrated that the HEMAcollect protein BCT samples consistently maintained hemoglobin levels below 100 milligram per deciliter concentration on the hemolytic index, indicating minimal hemolysis. In contrast, and as expected, 50% of the EDTA collected samples exceeded this threshold at the 7-day time point, as you can see here on the graph. Next, we looked at platelet activation, where we were really aiming to evaluate and demonstrate that the HEMAcollect protein BCT minimizes platelet activation. So we conducted ELISA-based analyses of key platelet activation markers, one being CD62P and another being EGF, which showed significantly lower concentrations in the HEMAcollect protein BCT derived samples compared to those collected and isolated from EDTA BCTs. And this really indicates limited ex vivo platelet activation in our HEMAcollect protein BCT, effectively maintaining EGF levels closer to baseline in the HEMAcollect protein BCT following exposure to those room temperature and simulated transport conditions, which we're showing the representative EGF graph here, and similar results were seen with CD62P. Next, we also really wanted to be able to demonstrate that the sample homeostasis is maintained within samples collected in the HEMAcollect protein BCT. So to do this, we evaluated two important protein markers, the first one being interleukin 8 or IL-8, which is typically released upon leukocyte lysis. And then we also looked at LOX1, which is a proxy we used as a proxy for protease activity. So LOX1 levels surged in EDTA samples, which really reflect high protease activity in that sample, where conversely in HEMAcollect protein BCT, we preserved LOX1 levels near baseline, indicating its ability to suppress protease activity, a critical need for proteomics discovery, where that uncontrolled degradation of proteins can obscure interpretations in the data.
Wow, that sounds really promising. Now, thinking about real-world use, is HEMAcollect protein BCT compatible with common proteomic platforms? And can you walk us through how it performed in both traditional and the newer high-throughput technologies?
Yeah, that's a great question. And we absolutely evaluated technologies broadly to ensure that real-world application to try and reach what researchers are doing today. So we looked at, in our performance evaluation studies for HEMAcollect protein BCT, broad compatibility with leading downstream proteomic technologies. So we looked at some more traditional based technologies, such as ELISA-based immunoassays, as well as mass spectrometry. So for ELISA's assays, the data that I just walked you through were actually conducted through ELISA-based assays, where plasma isolates from the HEMAcollect protein BCT were used to evaluate some of those critical markers of pre-analytical variability. So hemolysis for the hemoglobin concentration, the ex vivo platelet activation looking at CD62P and EGF, and then the blood cell protein release of IL-8 and protease activity through LOX1. So all of those were conducted with ELISA-based assays, and no upfront changes in processing steps were needed. Our samples were directly compatible with those assays. For mass spec, we conducted LC-MS assays to show and to evaluate direct compatibility. So in these studies, the HEMAcollect protein BCT derived samples were shown to be compatible with common upfront abundant protein depletion kits, as well as common LC-MS workflows. And they yielded a comparable number of detected proteins that were seen in EDTA controls. And our data suggests that HEMAcollect protein BCT may actually preserve proteins that are rapidly degrading in EDTA. And overall, the total number of proteins detected were found to be comparable, where our data actually indicated that there were some notable changes even within the 2 hours of processing where there was a change in abundance of targets in the EDTA samples that we didn't see in HEMAcollect protein BCT, which you can see here on the screen for the figure. So this was really important for us to show that we had that direct compatibility, and even highlights the potential advantage that our product would have over an EDTA collected sample. We looked at more high throughput, newer cutting edge proteomic technologies that are designed to be multi-target high throughput assays. So we looked at the Olink proximity extension assay, as well as the SomaLogic SomaScan assay for compatibility, as well as to evaluate the performance of our product more robustly in a larger targeted panel approach. So for the Olink proximity extension assay, or PEA, the HEMAcollect protein BCT demonstrated direct compatibility and superior stabilization compared to EDTA collected samples. Here we used an Olink 48 cytokine panel, and our results showed that 85% of proteins in the HEMAcollect protein BCT remained stable after 7 days of room temperature storage, compared to only 47.5% in EDTA samples. And even after simulated transport conditions, which is a more robust temperature challenge for the samples, the HEMAcollect protein BCT actually still maintained 80% of protein stability compared to just 32.5% in EDTA, as you can see here on the graph. And then when we were looking at the SomaLogic SomaScan technology, HEMAcollect protein BCT-derived plasma samples exhibited improved stabilization compared to EDTA-derived plasma on a custom 100 protein SomaScan panel. So after 7 days of room temperature storage, we showed that 93% of proteins in the HEMAcollect protein BCT remained stable, whereas only 76% remained stable in EDTA. And after those simulated transport conditions, followed by an additional room temperature storage of 7 days, the HEMAcollect protein BCT performed very well, maintaining 93% stability compared to 69% in the EDTA sample. And this data really demonstrates the robust performance and the direct compatibility of the HEMAcollect protein BCT on high throughput assays such as the SomaScan and Olink assays, with no upfront processing required to ensure compatibility.
Amazing. And then looking ahead, what impact do you anticipate on research environments and studies adopting this technology?
Yeah, this novel BCT with the ProteoPrecision stabilizing liquid it's specially formulated to preserve proteins and will allow scientists to advance proteomic research with greater confidence in the integrity of their samples from collection through to analysis. And we expect that the HEMAcollect protein BCT will support research goals by providing high-quality plasma samples by minimizing pre-analytical variability. And this will really enable the generation of reliable data through a broad range of downstream methodologies.
DNA Genotek develops products for high-quality biological sample collection. And to learn more about their latest innovation, the HEMAcollect protein blood collection tubes, request samples, or access resources, check out the links in the description. And if you enjoyed this episode, be sure to like and subscribe to catch all of our latest releases.